This video, I am going to talk about the eukaryotic replisome. Eukaryotic replisome. And so this is the complex of proteins that replicates DNA. So if this is, and you know what, I'm specifically going to focus on recent work that has succeeded, or apparently succeeded, in establishing the in vitro eukaryotic replisome. So, you know, all the proteins needed to replicate DNA in our cells were eukaryotes. So essentially the eukaryotic replisome that's in our cells replicating our DNA. Researchers have succeeded in establishing an in vitro version. So taking all the proteins, putting it in a test tube, and having those proteins replicate the DNA. And in a previous video, I alluded to how I think this is really, really interesting. You know, so PCR requires us to denature the DNA and use an enzyme that, you know, works well at high temperatures, and use primers. And but here, what they're going to be able to do, as long as we add a, you know, if we were performing the experiments ourselves, add a fragment of DNA that has an origin of replication, you know, the eukaryotic replisome will bind to the origin and copy the DNA. So unwind it. So bind, unwind, you know, make its own primers and copy the DNA. So really fascinating stuff. And let's see how this works. So this lecture is based off of a paper. You can check out the notes to get the reference of you know, the paper that this lecture will be based off of. Essentially, what researchers have succeeded in doing, starting with a DNA template that has an origin of replication. What they've been able to do is, you know, start with this template in, you know, a solution of water or buffer, add the ORC proteins, the CDC6 protein, CDT1, and the MCM2 to 7 proteins. Now, we saw all of these in a previous lecture. So these are all the components needed to form what? The origin recognition complex on the origin, and to install the MCM complexes at the origin. So then what the researchers figured out was they needed to add the DDK kinase. This is another thing we saw in an earlier lecture. So they add the DDK kinase at this step. It phosphorylates some of the proteins that have been established at the origin, that have bound to the origin. And then they add a couple more things, actually quite a number of things here, but we can summarize it by saying replication buffer, firing factors, and replication proteins. So the replication buffer, so what's in the replication buffer? Now this is important. In the replication buffer, they figured out we need to add deoxyribonucleotides. So deoxyribonucleotides, DNTPs, right? Because we're going to be copying DNA here. So the polymerases are going to be using the DNTPs to copy the DNA. But they also add ribonucleotides. Because just like in prokaryotes, how there is the polymerases need a primer, and the enzyme that makes the primer for DNA replication in prokaryotes makes an RNA primer. Same thing happens in eukaryotes. There's a polymerase, pol alpha, which makes the primers as part of the replisome. It makes an RNA primer, but instead of just being completely RNA, it makes about 10 nucleotides of RNA, and then, 20 nucleotides of DNA to follow that, or it might be the other way around. The exact number is not important. But we needed to add, there needs to be ribonucleotides. So we can abbreviate this as NTPs. So there needs to be ribonucleotides in the replication buffer. Now, what are the firing factors? We've seen a lot of these already. So the GINS complex, MCM10, CDC45. So we talked about these when we're talking about, activating the CMG helicases at the origin of replication. Now, there's a number of other things that we didn't talk about that are also needed. Pol epsilon. So this is one of the polymerases involved in replicating the DNA, but it's also required as a firing factor because, it might have a role in holding various proteins together. So a role independent of actually copying the DNA, just holding parts of the replisome together. And then there are 4 other proteins that we don't need to mention. And you can look up the details if you want in the notes. So when we add the firing factors, the DNA can be unwound, but it can't be copied yet. So that's where the replication proteins come in. So the replication proteins, now it's going to include things like RPA, replication protein A, and RPA is a single-stranded DNA binding protein. So you remember from prokaryotes have single-stranded binding proteins that stabilize the single strands of unwound DNA. Well, that's what RPA does in eukaryotes. They add topo 1 and topo 2, and these are topoisomerases. And we saw in our discussion of replication in prokaryotes how DNA gyrase was needed. Well, that's what topo 1 and topo 2 do in eukaryotic DNA replication. And we have pol alpha, which is the primase that makes the primers, and pol delta. So 3 different polymerases. And in an earlier video, we kind of covered the roles of these different polymerases. Pol alpha, making the primers. Pol delta, mostly lagging strand synthesis. And pol epsilon, leading strand synthesis. So that's the current model of replication and the major roles of these different polymerases. And there is an advanced, relatively new model where pol delta is involved in leading strand synthesis also, but we'll ignore that for now because it's a new model and definitely hasn't made its way to textbooks yet. Okay, so after this, what they do is essentially DNA is copied and they can analyze it by gel electrophoresis. Okay, so, it's understanding all these different proteins and their roles, it's not as important as understanding how they're able to, study the in vitro eukaryotic replisome. So we're really going to focus on how they're able to study it, how it works, how fast it replicates DNA, how long the leading strands are relative to the lagging strands, things like that. Okay, so when they go to analyze, let me diagram this for you. So when they go to analyze, so here's a summary of the processes I just went over. We just went over this, and I just copied this. So I just printed out the notes and copied this here. And I hope it's in focus. It's not really sticking down to the paper so well. But so they're going to look at the DNA either probably on an acrylamide gel. So this is agarose gel analysis. It's probably acrylamide, but it doesn't matter. It doesn't matter what type of gel they're using here. The concepts are going to be the same. So let's say they, you know, do this experiment here, and they go to analyze their DNA. They separate it by gel electrophoresis. The long products are going to be up here. The shorter products are going to be down here. And let's say they load the DNA sample in this lane right here. Well, you know, they might see DNA, and what you're going to see in a little bit is that they usually see long products and short products. I mean, that's what they usually get, but they can't see it, right? Because there's so little. They're working with such small amounts of the DNA template and the proteins that when they run the DNA on the gel, you really can't see it. Too little, I should say, too little DNA to see on the gel. So in order to do this, molecular biologists have to use a very, very common technique that allows them to see very small amounts of DNA after an experiment like this, after you separate it by DNA electrophoresis. We have a technique we can use to see DNA like this. And that technique is using radioactive deoxyribonucleotides. So let's take a moment to see how that works. So here is the generic structure of a deoxyribonucleotide that we should all know by now. This is something we should have committed to memory. So I'm putting 3 phosphate groups here. And the phosphate groups are linked to the 5-carbon sugar through the 5' carbon. And we should know all the numbers of the carbons because that's where, you know, this 5' to 3' directionality of DNA comes from, and it's so important in molecular biology. And this is where we'll have the base, A, G, C, or T. And there's the hydroxyl group down here connected to the 3' carbon. Now, for these phosphates have names. So this one right here, this is the gamma phosphate. This is the beta phosphate. And this is the alpha phosphate. So when a molecular biologist needs, you know, a DNTP, needs to incorporate say radioactive molecule into DNA, it's usually going to be P32, so phosphorus 32. Now, the most common form of phosphorus, the non-radioactive form, is P31. So P32 is the radioactive version. And what you should do is try to think about, okay, if you wanted an atom of P32 to be incorporated into a DNA molecule by DNA polymerase, which one of these should be radioactive? The gamma, the beta, or the alpha? Well, it should be this one right here, because when DNA polymerase is synthesizing DNA, these two are lost. And this one becomes part of the phosphodiester backbone. So by using DNTPs that have the alpha phosphate as P32, as the radioactive version of phosphorus, then a scientist can make the DNA molecules radioactive. Okay, what is the advantage of that? Well, okay, let's say, let me go back, where's that little... Okay, let's say a researcher does this experiment again, and in the replication buffer are the deoxyribonucleotides, and let's say one of them, let's say the DCTPs have P32 at the alpha position. Well, we do that experiment. and we load the DNA in here. Let's say in this well right here, we separate the molecules with a current. Shorter molecules down here, longer ones down here. And after we're done with the electrophoresis, we can't really see any of the DNA. But what we can do is take a piece of X-ray film or use a special detector called a phosphor imager that will detect radioactive decay. But X-ray film works just as well. So you put a piece of X-ray film over that gel, and the radioactive isotopes, P32, will break down. So when you develop the film, you might say, oh, okay, you'll see some dark spots on the film. And those dark spots, well, that's where the DNA molecules were. So using radioactivity, incorporating those into the DNA molecules, it allows scientists to detect very small levels of DNA. Okay, well, I hope that made sense to you. And you would use something like this when a DNA stain, say, wasn't going to be sufficient because you have too little, the levels of DNA are too small. So using a radioactive nucleotide allows you to see DNA when the levels are too low to see with a typical DNA stain. Okay, so now let's go back to the eukaryotic replisome here. One thing I wanted to touch on, when radioactive nucleotides break down, they release particles like beta particles and alpha particles and things like that. And that's what the X-ray film is picking up, or the phospho imager will be picking up as the signal. Now I'm trying to find my notes here. What are we looking at? What do we want to do? Okay. So I want to go back to this. Okay, so where were we? Now, when the researchers started working with the eukaryotic replisome and they're using their radioactive nucleotides, what they were seeing was, what I already alluded to, that the eukaryotic replisome was making DNA of, approximately 2 different sizes. So long products and short products. And they hypothesized that the long products were a result of leading strand synthesis. And the short products were a result of lagging strand synthesis. And if you're confused on, what is leading strand synthesis, what is lagging strand synthesis, make sure you go back and watch the video on 6 steps of DNA replication, and that should be in the Lecture 12 playlist, Lecture 13 playlist. And okay, so this is the hypothesis, that the long strands are a result of leading strand synthesis and the short strands are a result of lagging strand synthesis. And that kind of makes sense with, what we already know, but they couldn't be sure. And they had to test this hypothesis somehow. And they did that in a really neat way. And I like talking about this experiment because it brings in, a lot of different concepts, major concepts in molecular biology with respect to DNA replication and 5', 3' orientation of DNA molecules. Now we have radioactive nucleotides, so a lot of really important stuff are involved in this experiment. So the DNA template they're going to use to test this hypothesis looks like this. So a linear DNA molecule, and there is going to be a yeast origin of replication, so the autonomous replicating sequence, ARS, and this is This is about 100 to 150 base pairs long. And then over here on this flank, this strand up here is going to be T-rich, and this strand down there is going to be A-rich. Over here, A-rich up top, T-rich down bottom. And in the notes, you'll see there's like a magnetic bead here. We can just ignore that. Doesn't really influence the experiment at all. This flank over here is 1.4 kb long. So what does that mean? That's 400. Kb is for kilobase, so 1400 base pairs. Same thing over here, 1.4 kb long. So it's a relatively long DNA molecule. And let me, just to make sure you understand, so essentially when I say A-rich, it just has lots of A's. And this is a bit of an exaggeration, right? Because there are some C's and G's and maybe even a few T's in here. But for the most part, it's mostly, A's. Then we have the ARS sequence here. And then we're going to have a bunch of T's over here. Again, this is an exaggeration. There are going to be some C's, some G's, maybe even a few A's. But for the most part, mostly T's. And I can put the complementary strand in down here. Okay, so we have this DNA molecule, and this is going to be the template for this experiment right here. And what we're going to see is that this template right here, when it's replicated, the leading strands, remember the leading, we use leading and lagging to refer to the newly copied DNA, to the newly synthesized DNA. So the leading strands should have mostly T's, and the lagging strands should have mostly A's. And this is going to allow them to test their hypothesis, right? That those long DNA molecules are a result of leading strand synthesis, and the short DNA molecules are a result of lagging strand synthesis. So picture this molecule right here. Now, this is the origin of replication. So the replication forks are going to start here. One's going to move that way. One is going to move that way. And I could just diagram that roughly like this. So two replication forks. We can divide this in half. This is the T-rich template. This is the A-rich template down here. A-rich right here. T-rich right here. 5' end, 3' end, 5' end, 3' end. Okay. Now, this is really messy. And it's kind of confusing to consider both replication forks, right? With two eukaryotic replisomes, one moving to the right, one moving to the left. Kind of hard to consider all of that together. So what we're going to do, I'm going to split this right here and do one replication fork at a time. So let's take the right half, the replication fork on the right. So it's moving this way. And here's the T-rich strand, which is going to be the template for the lagging strand synthesis. And here's the A-rich strand, which is going to be the template for leading strand synthesis. And how do we know that this is the leading strand? So take a look at the 5s and the 3s. So the 5' end of the leading strand is going to be here. It's going to have to go this way. I'm not going to diagram the primers. The important thing is to just know that this is the leading strand. And why is it leading? Well, it's moving in the same direction as the replication fork. And this is going to be the lagging strand. How do we know that? Well, one way we know that is it's moving in the opposite direction to the replication fork. And I didn't put the primers in. I didn't put in the fact that all models pretty much say the lagging strand is made by Okazaki fragments. But what we can see is that the leading strand is going to be mostly T's, right? Because the template's all A's. the template is mostly A's, so the leading strand is going to be mostly T's. And the lagging strand is going to be mostly A's. So in summary, you know, replication in this direction is going to lead to leading strands that are mostly T's and lagging strands that are mostly A's. Now we can see the same thing with the other replication fork. So here's the 5' end, 3' end here, 3', 5' here. Remember the T-rich strand was on the bottom, the A-rich was on the top. So leading strand going in the same direction as the replication fork, mostly T's. Lagging strand going in the opposite direction as the replication fork, mostly A's. So, okay, again, leading strand, T's, lagging, A's, just like at the other replication fork. So now the researchers have a hypothesis. Let's review their hypothesis that the long products made by their in vitro eukaryotic replisomes are a result of leading strand synthesis. And the short products are a result of lagging strand synthesis. And the experiment is designed with that template, right? I should put the template here. This template with A-rich strand there, T-rich here, A-rich here, T-rich here, is designed so that the leading strands are mostly T's and the lagging strands, I should say are T's, I should say contain mostly T's, contain mostly A's. Okay, so it's a great experiment to test this hypothesis. Because what are they going to do? Okay, so one more thing I have to add to this experiment. So they add this template here, which is the DNA template. They do this experiment. And in the replication buffer, they're going to use two different radioactive nucleotides. So in one experiment, they're going to have A's, C's, G's, and T's, but the T's are going to be radioactive. And in the other, I guess the other assay, I call this an experiment, in one assay, in one or reaction. In the other reaction, the A's are going to be the radioactive nucleotide. So when they do the experiment and they get the results, what they see is that they can see DNA up here for reaction 1 when the T's were radioactive, and they can see DNA down here for reaction 2 where the A's were radioactive. Now this result is consistent with the hypothesis, right? So they set up the experiment, they know the leading strands, they set it up so the leading strands will have mostly T's. And when they have the T's as the radioactive molecules, they only see DNA up here. They don't really see any down here. It's because, you know, the short strands, which are predicted to be a result of lagging strand synthesis, don't have any T's. The same thing over here, right? They set up their experiment so the lagging strands would contain mostly A's. And when they made the A, the radioactive nucleotide, they only see, okay, yeah, sure, they only see DNA down here. Is there DNA here? And is there DNA here? Yeah, there's lagging strands made in reaction 1, but the lagging strands don't have any T's. So they don't see this DNA. There's no radioactive signal coming from here. Same thing for this. There's leading strand DNA being made in reaction 2. There's just no A's in there. So we don't really see any DNA over here either. So it's there, we just don't see it. The radioactive nucleotide that was found in both strands, well, we would see signals at both. But the experiment was designed purposely so you'd only get one or the other. If their hypothesis was false, you know, if leading strand synthesis resulted in long and short products, well, then we would see DNA signal coming from both. Same thing for lagging strand. If lagging strand synthesis made long and short products, we'd see both. But that turns out that's not the case. Their hypothesis seems to be correct. The long products were a result of lagging strand synthesis, and the short products were a result, the long products were a result of leading strand synthesis, and the short products were a result of lagging strand synthesis. Which is great, you know, because it fits with our model of, you know, how replication works in vivo, with lagging strand synthesis being a result of, you know, Okazaki fragments, short Okazaki fragments needing to be linked together. So, okay, that's great. That's a really neat in vitro experiment, and the replisome seems to be working.